How do you keep and share your most important memories? For many of us, it's those pictures we take on our phones and share through email and social media. But as some of us age, the memory systems in our brains will begin to slip and become unreliable. This can be the beginning of conditions like Alzheimer's disease and dementia. People with these ailments will begin to forget things. They can get to the point where they no longer recognize people they've known for decades, even their spouse and their children. Researchers at Arizona State University are now at work on several new ways to unravel the questions around how Alzheimer's begins in our brains and possibly ways to slow or even stop the disease.
Alzheimer's disease is not just aging. There's a significant difference between aging and Alzheimer's disease.
Researchers are grateful to have a powerful tool: volunteers.
We study people during life and after death. And some of these people have been coming in and seeing us for 20 years before they die.
It is these people who have allowed thousands and thousands to understand disease. Without them, this wouldn't be possible.
Researching Alzheimer's, next on Catalyst. This YouTube edition of Catalyst is brought to you by Arizona PBS and Arizona State University. Life has problems. Science turns them into questions that can lead to solutions and even innovations. This is Catalyst, shaping the future through science research at Arizona State University. At the end of life, most of us feel one of our last decisions needs to be what will happen to us after we're gone, a choice between being buried or being cremated. You'll also hear of people who've decided to donate their body to science after they die. Now, medicine can really learn a lot from people who do this. And in the search for answers to Alzheimer's disease, some people are making an even more unusual choice. They're letting their bodies be studied for signs of the disease both before and after they die.
And number 2 is actor Barrymore. It starts with an L.
Lionel.
Sandy and Harry Abramowski enjoy crossword puzzles, but for Sandy, they're getting harder.
Her short-term memory is virtually non-existent. You can ask her what day it is, and she probably won't know.
It's not something that came on like that. It's something that comes on slowly. And then you start thinking like, I don't know if that's right or wrong. I better ask Carrie.
She'll recognize her granddaughters and grandsons, but she may not remember their names anymore.
I'm not capable of necessarily making the right decision. And if I say, Harry, I want to do this, he might say, Sandy, that's not the way to go.
Even before Sandy was diagnosed with Alzheimer's, we felt that we might as well donate our bodies and maybe help somebody else.
If I'm going to die, it isn't going to help me any. But is there something in there that might help someone in the future? We don't know that.
I'm Tom Beach, and I'm a neuropathologist, which means I diagnose brain diseases under the microscope. I'm the director of the Brain and Body Donation Program, which means we follow older people during life annually with neuropsychological tests and movement tests, and then when they die, we do autopsies and sample the brain as well as all the body organs for research use. Okay, I'm going to show you the difference between a normal brain from an older person and the brain of someone who had Alzheimer's disease. I'm going to lift the brain out. This is of the normal person. It's a normal older person. I think they were 64 years old at the time of death. And you see all of these squiggly things on the surface of the brain are the gyri of the cerebral cortex. And you see how full they are and pressed together they are. And I'm going to show you now a brain from someone who had Alzheimer's disease. And it's only half of a brain, but I think you can see the difference in that the gyri are much narrower and the spaces between them are much wider. And in the normal brain, the ridge tops are broad and the valleys between them are very narrow, but in the Alzheimer's brain, the ridge tops are narrower and the valleys between them are much broader. This is what results when many of the brain's processes die back. The nerve cell's branches become pruned back because the nerve cell is sick and it cannot maintain all those branches. During life, the diagnosis of Alzheimer's and Parkinson's is really a best guess by the neurologists, and it's fairly accurate, but for research purposes, we want to be completely accurate. So at autopsy, we confirm by looking in their brain under the microscope whether they really did have Alzheimer's and Parkinson's. By analyzing the chemistry of the brain, hopefully we can identify the first 2 or 3 chemical reactions that started off the disease process. And if we can identify those chemical reactions, then we can think of drugs that could go into the chemical reaction and change it so that the disease does not progress.
For all of our donors, their donation follows a standard set of procedures. We have to go from the whole brain to something that's sliced very thinly and stained on a glass slide. The type of microtomy I'm doing here is called sliding freezing microtomy, which involves freezing the tissue and using a device to cut the frozen tissue such that we get those sections, and which can later be mounted on glass slides. Each of these brain slices is pretty large, and some of the brain regions we look at are pretty small.
Our brains have many connections to and from other parts of our bodies. That's why researchers are also preserving and examining other major organs along with the donated brains.
And I'm pulling out a heart now from a recent autopsy. This is undissected.
Here, inside the hearts of Alzheimer's disease victims, a contradiction. Their brains may be ravaged, but their hearts, they often seem healthier than the hearts of many older people who don't have Alzheimer's.
So is the combination of heart and brain disease additive or is it synergistic? We still don't know. But we have published a study on the heart in Alzheimer's compared to the heart in normal older people. And surprisingly, the heart in Alzheimer's seems healthier. They're about 100 grams lighter on average. And for a heart, it's good to be lighter. And they have fewer diagnosed cardiovascular conditions. And it's probably because when people are coming down with Alzheimer's and becoming cognitively impaired, they begin to lose weight. And so on average, our Alzheimer's patients have a BMI of about 1.5 points lower than the normal elderly of the same age. Many older people do eventually get Alzheimer's disease. So if we're following 400 or more annually, some of them inevitably come down with Alzheimer's disease. So looking backwards in retrospect, we can see if there was anything about their clinical testing that might have given us a clue that they were about to come down with Alzheimer's, because our focus and the focus of much of the research world in Alzheimer's and Parkinson's is trying to detect people with disease before they even know they have disease so that we can use treatments to prevent further deterioration.
Right now, the only sure way to know exactly how Alzheimer's and dementia have changed someone's brain is to carefully examine that brain after death. That makes it hard to find useful treatments for the person suffering the disease while they're still alive. So scientists are spreading out into new places looking for clues. Things like family history, diseases earlier in life, and roles played by the different kinds of cells we have in our brains.
Alzheimer's disease is not just aging. There's a significant difference between aging and Alzheimer's disease.
Diego Mastroeni is one of the scientists inside the ASU Banner Neurodegenerative Disease Research Center at Arizona State University. Alzheimer's disease begins when people start to get an abnormal collection of proteins building up in their brains. One is called beta amyloid. It forms plaques on the outside of brain cells. Inside those nerve cells, another protein can form. It's called tau. Tau begins to make what researchers call tangles. Over time, the buildup of these plaques kills nerve cells in the brain. Memory grows weaker, dementia begins. Whole regions of the brain may start to shrink.
What we do, pretty standard for brain pathology and to determine whether someone has Alzheimer's disease or not, we'll take a human brain who is greatly donated by an individual who comes to autopsy. And what we like to do is we will then take a slice of the brain. So we would actually cut the brain here and we will take it onto a microtome and cut it even thinner. Now we can do our stains and stain for things like amyloid plaques or neurofibrillary tangle, the kind of pathological hallmarks of Alzheimer's disease. So this is the brain, slide, and then we'll go on to looking at it under the microscope. Okay, so now we'll take our brain tissue and we'll come under the dissecting microscope where we will identify and make sure that we have the appropriate brain region that we want to cut thinner to do some sectioning. So from the whole brain chunk that we took off, now we can actually section it into a very smaller, really thin sections. These are approximately about 20 microns in diameter. That is thinner than a piece of paper. And now we will take it through the staining procedures. So now the brain is sectioned thin enough where we can stain it, and we have stained it with the silver stain, which is a marker of the pathology that's associated with Alzheimer's disease, which is neurofibrillary tangles and plaques.
Neurons send commands to our muscles. They transform and relay the electrical signals that control our movements and the function of our bodies.
So the major areas of interest in Alzheimer's disease is the limbic system, but in this section we look interested in the neocortical regions. This is the frontal cortex, so we are interested in these regions right around here where it's really neuron dense. You can see over here is a lot of white matter, which is neuron non-dense, which is a lot of axons and processes go through. So the major focuses which I'll be showing you will be looking at these first 4 or 5 subregions of the neocortex. So from the stained tissue section, we are now going to look at whether this person has Alzheimer's disease or not. And as you can see from the image here, and this is the neocortical region I was talking about in the whole brain tissue, you can see that there's a significant amount of plaque pathology, and this individual would be positive for Alzheimer's disease. So what we're looking at here is these are really dense core plaques in the Alzheimer's brain. And if it gets to this point, I think it's too much. So what we're focusing on are these little spots here, these little brown spots where these are like the oligomeric forms of amyloid beta before it gets into these really large aggregates.
One of Mastroeni's big ideas is this. Alzheimer's shows up later in life, but we need to start looking for signs of Alzheimer's earlier in life, for clues and perhaps even cures.
For this particular study, what we were looking at was neurons. So the problem is all these other studies have been done for decades now, have taken whole brain, what they call homogenates, where they grind all the brain together. The problem is that the brain has many cell types, many cell classes. So we didn't know from what cell were these changes specifically coming from. This is the reason why we use the laser capture microdissection system, because we can isolate specific cells. So in this case, we isolated neurons, specific in an area of the brain that's affected in Alzheimer's disease, which is a CA1 of the hippocampus.
So right here we have a section of hippocampus on a slide, and this is on the microscope, so we could view it through the microscope, but we can project it onto the screen, and then we get a large picture of what we're working with in order to do the actual laser dissection. And these are the pyramidal neurons in the CA1, and so we're just going to increase magnification to 10x. And so you can clearly see pyramidal neurons here, and the nucleus is very obvious in each of these neurons. And so we want to cut as closely to the nucleus as possible. So I'm going to go ahead and circle just around the nucleus. And depending on the study, you know, I might need to get 500, or I might just need to get 50, and sometimes we even look at a single cell. So this is a section that has already been cut. So the neurons have been removed from this.
So once we were able to look at these specific cells that get really sick in Alzheimer's disease, we got a lot of clarity in terms of specific cellular changes that now we can address. And what we found was that we found many mitochondrial issues, both nuclear and mitochondrial encoded problems. And we were able to isolate those using this kind of technology. Mitochondria are the powerhouse of the cell. They give the cell all the energy that is required.
They produce about 90% of the chemical energy that cells need to survive.
So what we're trying to do is we're trying to protect the mitochondria. So to protect the mitochondria from oligomeric amyloid beta, we're using these coenzyme Q10 analogs to protect them, to give them the energy and the power to fight off disease.
Coenzyme Q10 is an antioxidant that your body produces naturally. Your cells use it for growth and maintenance. The levels of coenzyme Q10 in your body decrease as you age.
Our body makes so much coenzyme Q10 on its own. It makes 99% of it on its own. 1% of it we get from diet. As we get older, and we only make 75% of it, and we can only still get 1% from diet, we have to have some kind of supplementation. Over, let's see, 10, 20 different studies now have shown that mitochondria are affected early in life, early changes in Alzheimer's disease and also in aging. So the different mechanisms by which we would try to do these kinds of things would have to be early in life. And one way we're starting to work on is on a blood test as a predictive measure. Everyone's at risk for developing Alzheimer's disease. So think of it as a scale. So are we at less risk or at more risk? So these individuals who are at more risk would be able to be detected with our blood test. And if that was the case, it gives you a percentage. Say, I'm at 80% risk. Can we reduce this risk of Alzheimer's disease using these kinds of treatments like the coenzyme Q10 analogs that we're trying to do? So that's basically the purpose. So early detection and early treatment.
The work done by Mastroeni, Beach, and other scientists creates an enormous amount of data. Data that includes a patient's DNA, protein levels, clinical observations, and more. Researchers extract valuable information from the patterns they see in these large data sets, and it's giving them insight into the common traits found in the brains of Alzheimer's patients.
When an individual chooses to donate their brain and their body to a brain bank or program like the ones that we've benefited from, investigators will take samples from different brain regions and then perform different sorts of what's called molecular profiling. And so the brain is made up of a universe of cells, different cell types. And then within each cell, you have your genome, which is your DNA. And this is a template that tells your body, tells your cells how to act. So we want to measure the DNA, the genome, and then we also want to measure how much all the genes are switched on or off. And that gives us something like a snapshot of what that cell is doing at that moment. We also want to know what sorts of neuropathology were present in that person's brain. Are there tangles? Are there plaques? How are they distributed? How severe are they? And we also want to know how severe was that person's clinical dementia. And so all of these different ways of describing this same system become part of these huge data sets, essentially, that we were able to leverage in this study. As we kind of began that analysis and dug into our results, what we began to see were clues from a number of different directions that we thought were suggesting that maybe some kind of viral activity might be explaining some of what we were seeing. And so we had early access to the Mount Sinai Brain Bank data that had been collected. And so what we were able to do was then to take a data-driven approach where we looked for all viruses that are known to infect humans as a primary host. So this is over 500 viruses. And then just simply see if we saw any evidence for viruses. The first point where we really began to think we might be onto something was when we looked across all of these different samples and we saw that there was an increased abundance of two particular human herpes viruses. There's a human herpes virus 6A and number 7, HHV 6A and 7. And these are very common pathogens, and so we weren't really sure what to make of that. One of the things that we were able to do, though, by virtue of having access to these huge biorepositories, we were able to actually replicate this finding in 2 additional cohorts. One of the things we did see emerge from our study is that as the number of viral particles that we were detecting in the brain increased, the number of neurons that we would detect in that same brain region would decrease. These are viruses that are very, very common. So in the United States or in the United Kingdom, probably 9 out of 10 kids are exposed to these viruses within the first few years of life. They're actually typically thought of as fairly mild viruses. They cause rash, fevers, and tummy upset in young kids. We're mindful that some people might find these sorts of results disturbing or alarming. And that's certainly not our intent in talking about these findings. So we wanted to emphasize that what it is we've found doesn't change how it is we'd go about examining an individual person's risk for the disease. The fact that someone may know that they've been infected with these viruses in the past doesn't mean that they're going to get Alzheimer's disease. These are such common pathogens that there's many more levels of complexity that we need to understand before we know how a virus that is so common seems to become abundant in some people's brains, but not in other people's brains. So what we're reporting isn't anything that would impact clinical care at this stage, but what it does give us, I think, are a set of particular hypotheses that we think deserve careful attention. In some ways, it's been kind of a controversial idea. The really tricky thing with something like what's called the pathogen hypothesis of Alzheimer's disease is really working out whether or not it actually represents a causal contribution, where the viruses or some pathogen might actually be contributing to the risk of the disease or maybe accelerating it once it's established. But, the very real alternative is maybe these are just species that are given space to emerge and, in the presence of neurodegeneration that happens due to other means.
As scientists continue their research on the disease, Reedhead's work will help scientists better understand the possible ways that Alzheimer's emerges in the brain.
My grandfather passed away with Alzheimer's disease, and at every stage of kind of watching him go through it, there's kind of a new layer of tragedy that kind of unfolds, kind of as you watch what someone else is experiencing, their subjective experience of feeling important parts of themselves slip away, and then you see the kind of the ripple effects on family and friends and loved ones, and then and I think there's also just a huge loss to the community in general. The price that this disease exacts in terms of just the loss of life experience and wisdom from kind of seasoned members of the community. Everyone loses from that.
If someone you love suffers from Alzheimer's, or you would like more information about the early signs of the disease, go to alz.org. That's the website for the Alzheimer's Association. I'm Professor Vanessa Ruiz, and what you've been watching is Catalyst, our show about shaping the future, how research creates real-life results. And because our lives always have new problems that science can help to solve, we'll be back soon with more stories.
Catalyst is supported by Knowledge Enterprise Development at Arizona State University, advancing entrepreneurship, innovation, discovery, and knowledge for the public good.
Subscribe to this channel to see more episodes of Catalyst on YouTube.